Hello everyone, myself Bhumika Naik. I am student of Ssjcop College of Pharmacy. I am studying in final year of B Pharm. My today's topic is raw material analysis under the guidance of Dr. Nitin C. Moire sir, who is a professor and principal of our college. Content: Introduction, importance of raw material testing for pharmaceuticals, need for raw material testing, raw material testing. First, the introduction of raw material analysis. Quality assurance and quality control are the important in the pre-production stage of pharmaceutical production. So, to verify that the raw material do not contain any impurities that can cause the health hazardous or more complex purification and lower yield. Raw material ID is the process of evaluating these raw materials to identify and discard any impurities or adulterants that could have any adverse effect on the final product. In raw material analysis, a variety of QA and QC analytical instruments to perform raw material ID using a variety of different techniques. Importance of raw material testing for pharmaceuticals. Raw material testing is crucially important for ensuring safety, quality, and efficacy of pharmaceutical products. There are many things to be considered that could impact the way raw materials need to be blended, such as polymorphism, the particle size of the raw materials, or other properties. Hence, the raw material analysis is essential to determine the purity, identity, and quality of the raw materials before they go into manufacturing process. Need of raw material testing. As hundreds of raw materials and ingredients are used in the process of formulating the final pharmaceutical product, it is quite tough to check every ingredient for quality. Unless the ingredients have undergone quality testing beginning the manufacturing process won't be possible. Moreover, if the low quality raw materials are used, it will result in a low quality finished product which could face product recall. This can cause significant damage to material cost as well as reputation. Therefore, raw material testing in pharmaceutical is necessary. First need of raw material testing is ensuring a quality product. Pharmaceutical raw material testing is carried out established that all the incoming raw materials match the right specification and requirements. Needless to say, incorrect supply of raw materials will lead to compromise in the safety and quality of the product. Besides, it will also manufacturing delays and significant wastage of time and costs. Therefore, testing lab help in pharmaceutical companies lay down the specification of the raw material right from the initial stage of drug development. Second need is testing lab standard and approval needed. Every pharmaceutical product or medical device has to be approved by the state FDA and the Central Standard Control Organization, that is SOFTCOPY, before it rolled out for public use or commercialization. Testing laboratories can help carry out the material analysis, DSC analysis, chemical test, physical characterization, NMR testing, FTIR testing, and more. All according to the specification and safety protocols established by the FDA and SOFTCOPY. Traditionally, chemical testing laboratories perform the raw material testing and prepare the reports to determine the quality and suitability to be used in pharmaceutical drug formulation. They are well equipped to carry out the sophisticated procedures involved in the raw material tests. Next is raw material testing. Quality control raw material testing. Before manufacturing begins, all the materials must be tested for purity, identity, and quality, depending upon the type of product, tablets and capsule versus biotic product. As few as 15 to 20, as many as 60 of raw materials might be needed for the product development. The extent of raw material testing is determined by the manufacturer. A conservative approach will be perform complete analysis of each lot of raw material received. Pharmacopoeia provide monograph for the most commonly used raw materials in the pharmaceutical industry. These monograph details several different analytical techniques. Fischer moisture analysis, pH, viscosity, and titrations are the commonly, but are the more complex techniques such as HPLC, GCMS, and ICP-MS are the sometimes required. The analytical chemistry and microbiological teams can help you with the necessary testing of raw materials, APIs, finished product, packaging material, and medical devices. Testing services for raw material is physiochemical properties, identity and purity of small molecules, identity and purity of large molecules. Physiochemical properties testing services. Testing services for physiochemical properties include appearance, pH, moisture content, osmolality, viscosity, optical activity, and spectral analysis. Appearance. This ensures that all tested solutions are essentially free of visible particulates, physical state, color, odor. pH. pH will determine whether the sample's acidity or alkalinity meets the client expected specifications. The pH of small molecules, pharmaceutical or biological formulation can significantly affect its stability, solubility, and in vivo delivery, making this test a crucial element of any lot release program. Moisture content. A large variety of pharmaceutical and biological products contain water as hydrates or in absorbed form. The degree of water content can influence the therapeutic shelf life, activity, and quality, thus the requiring compliance to be predetermined specifications. Osmolality. Osmolality is the another important factor of lot release for a development drug, and it is defined as total number of solid particle per kilogram of solvent. Freezing point of technology to test the osmolality of prepared solution to be ensure correct isotonicity, consistency, formulation, and product stability. Viscosity. A fluid's resistance to flow, measured a viscosity, is a principal component of lot conformance. Viscosity is a fundamental to the formulation's intended progression and activity and may be used as a benchmark for compound concentration or degradation. Optical activity. Many pharmaceutical compounds are chiral and hence optically active given that they rotate plane polarized light. The major optical activity of the provided solution can then be used by the client for many different aspects of lot release program. By comparing with predetermined target specification, polarimetry may be used to detect impurities or to determine the concentration of the intended chiral product. Spectral analysis. The unique absorption spectra of chemical or biochemical products can serve as a distinguishing physiochemical property of pharmaceuticals and biologics. Analysis employs ultraviolet visible and Fourier transfer infrared spectroscopy for target compound identification and concentration measurement. Spectral analysis may also be used to reveal impurities in the yield product. Identity and purity of small molecules. Small molecules identity and purity testing. Identity and purity testing are crucial requirement of lot release testing services for identity and purity. HPLC, high performance liquid chromatography with different detectors. Mass spectroscopy, LCMS, LCMS or MS, and TOFMS, UV visible, ultraviolet visible spectroscopy, FTIR, Fourier transfer infrared spectroscopy. HPLC, high performance liquid chromatography. Chromatography describes the use of high performance liquid chromatography for qualitative and quantitative analysis. This is done by comparing the chromatogram with the reference peak. Discrepancies between the expected and experimental chromatograms may indicate source of impurities within the lot. Mass spectroscopy. LCMS, liquid chromatography mass spectroscopy, is an accurate and reliable method of chemical analysis that combines the advantages of liquid chromatography and mass spectroscopy. Mass to charge ratios derived from the analytical technique can be reveal molecular weight of each compound and therefore help with the identification of the analytes and impurities. UV visible, ultraviolet visible spectroscopy. Spectrophotometric tests are critical for the identification of many chemical substances. These tests yield absorption spectra that demonstrate rough identification of the formulation and any contamination. FTIR, Fourier transfer infrared spectroscopy. Infrared spectroscopy is normally used the most powerful test in confirming the identity of small molecules and pharmaceuticals. FTIR result may use to compare multiple specific peak in the IR spectrum with those of the reference standards, establishing a robust acceptance criteria. Identity and purity of large molecules. Large molecule identity and purity testing. When manufacturing biological products, large molecules in the imperative that each lot produced conforms to predetermined specification. Testing services for identity and purity, HPLC, high performance liquid chromatography, mass spectroscopy, Western blot, ELISA, that is enzyme linked immunosorbent assay. Mass spectroscopy. Mass spectroscopy can be used to identify biologic product and alter the manufacturer of any impurities. SDS, PAG, sodium dodecyl sulfonate polyacrylamide gel electrophoresis. SDS PAG separates out the protein in the polyacrylamide gel based on size. Western blot. Western blots are conducted by first running an SDS-PAGE gel and then transferring the proteins onto a membrane. This method can be used to identify the protein product or product related to impurities. ELISA, enzyme linked immunosorbent assay. ELISA is used to detect proteins and determine the concentration of the protein interest. Indirect, direct, and sandwich ELISA are the most common variations of ELISA. The absorbance reading from each well are compared with the standard curve to determine the concentration of the protein of interest from each sample. Thank you.